Hi everyone, so we're going to pick up where we left off. We're going to talk about the medulla, which is the central portion of the hair shaft. And so what this is a collection of cells that resemble a canal, and that runs along the length of the hair. So there's also such things as the medullary index. So not only do we have different types of medulla, we'll look at that in just a moment, we also have what's called a medullary index, and this is where they can measure the diameter of the medulla in relationship to the diameter of the entire hair shaft. A general rule of thumb is that animals have a medullary index of 1/2 or greater, whereas humans have a medullary index of 1/3 or less. I would love to do a lab activity where you guys could measure the medullary index, but unfortunately, I don't have the type of measuring tools that we would need to make that successful. But in general, that is something that you will need to know for your test, so please make note of that. Okay, so the shape of the medulla changes from animal to animal, and humans have cylindrical medulla, if they're present at all, whereas cats have what looks like a scaly-looking medulla. So just like with the cuticle a few minutes ago, the medulla can also be unique to the type of species that a hair sample that we're observing. And another thing of importance that I spoke about just momentarily that I'm repeating again and we'll talk about in a moment is that your own hair, let's say that 3/4 of your hair have continuous medulla. Oddly, not all of your hair samples will have continuous medulla. So the point is, I said that backwards a bit, is that you may have a large portion of your hair samples that are indicating that you have continuous medulla, but you will also have hair samples within your scalp that will not show that same feature. So again, reiterating that hair is trace evidence. It helps us go back to a group of individuals. It doesn't necessarily help us individualize. Okay, so there actually are 4 types of medulla. The 4th one's not pictured because the 4th medulla is absent. So that means there wouldn't be one. So we have continuous. So that is where you have a line that continues throughout the shaft of the hair. You have what's called interrupted and fragmented, and there's a huge difference between the two. Interrupted medulla is where it breaks off from creating a continuous line, but how it does it is generally the same. Okay, so you're seeing a repetitive pattern within a same amount of space. Whereas with fragmented, that you have an unevenness of whenever that medulla fragments or is missing a piece of that continuous line. So, and again, so the last one is absent, so that's what our 4th one is. And the 3 forms vary from person to person and even from hair to hair on a single individual. Okay, so just a couple of pictures of different type of medulla. You can see here the one on the left. I hope that you would choose, go ahead and think about it, fragmented. That's exactly right. The center one would be absent, so there is no medulla. And the one on the right, which looks like that pencil from several slides ago, is continuous medulla. And you can see here the one on the right. And now, of course, we have some things against us right now. We don't really know what magnification settings that we're looking at each of these samples, but I may be inclined to believe that the one on the right would be from an animal species, based on the medullary index characteristics that we just discussed about some being 1/3 greater or 1/3 less the size of the diameter of the hair shaft. Okay, so we want to move into the roots. Why do we want to talk about roots for forensic science? It's really important because root of the hair changes shape based on the phase that your hair growth is in at that moment. And also another thing that's important for you to realize is that not every head of hair on your head is at the same growth stage. And if it were, that means that you'd lose all of your hair at the same time and have to regrow that. So we have millions of follicles on our scalp, and they each run independent of the other. So you're going to have hair on different parts of your head in different growth phases at all times. Thank goodness, right? So The hair root allows for the growth of hair, and there are 3 phases that you move through. You have anagen. I'm sorry, I'm laughing at my daughter. She's crazy. Anagen, catagen, and telogen. So that's ACT if you take and make an acronym of that, and that's ACT. So ACT, and that would be the order. Anagen being the first phase, catagen being the second phase, and telogen being the third phase. Each root phase has a different shape and size, so yes, it is useful potential forensic evidence. As well, the most important part that we would want to get from this is that we're looking for the potential of the presence of a follicular tag, which is where those dead skin cells that have been removed with the hair, because the follicle and the root are still attached to one another. So that would be in your anagen phase and a little bit of your catagen phase. Okay, so here's just a couple of pictures showing you some different phases. So it's really kind of cool that the follicle actually just moves and changes with your hair as it's moving from one phase to another. And the purpose of that is that it starts out big and healthy, and then as your hair gets older and older and you're moving from one phase to the next, the follicle is changing in size and shape to help get rid of that hair sample so that it can start anew and grow a new piece of hair. Okay, so the anagen phase. So the anagen phase is our first phase. It is also our longest phase. So keep those 2 things in mind. This is stuff that you will need to know for your test. Can last up to 6 years, which your hair is in continual growth throughout this anagen phase. The root is attached to the follicle, and it resembles what looks like something on fire, a flame. I almost think of that as like it's a matchstick. So whenever hair is forcibly removed in the anagen phase, we will generally have the follicular tag, which means we generally have DNA evidence that we can now use to individualize that hair sample to someone. Our second phase is the catagen phase. So the catagen phase can last 2 to 3 weeks. This is the second phase. Your hair growth slows and eventually ceases to grow, and it's getting ready for that third phase to push it out. So this is whenever the root of your hair, the root starts taking on this elongated appearance as your follicle is trying to readily push that hair sample out. And our last phase is the telogen phase, anywhere from 2 to 6 months in length. And so what happens is that the root of your hair takes on this club shape, and hair growth is completely stopped at this point. And the telogen phase, remember, is our third phase. So the hair is eventually forced out of the follicle, and it's shed naturally. Now, that's happening every single day. And ladies, you know what we're talking about, because generally with that longer hair, we see that hair more readily, whereas gentlemen that typically have the shorter hair. So a lot of times you don't notice, your hair falling out as frequently as a female may notice hers. Okay, so forensic analysis of hair. So the following questions apply to hair evidence. Is the hair human or animal? Does it match the hair of the suspect? Does it have a follicular tag for DNA testing? And we're going to go ahead and stop here, and we'll pick up with notes later. Thank you.